Okay, so there'll be a link to this, I think, if there's anything that I say that you want to go back to or see again, I guess you'll have access to the recording. So I just take a couple seconds to introduce myself. So my name is Kathy Royer, and I'm a faculty member in biological sciences at RPI. And I'm interested in proteins, so that's what I've kind of prepared, but not just any proteins, proteins that come from organisms that live in really strange environments on Earth. So that's what we'll talk about today. Maybe you could tell me a little bit about yourselves.
My name is Mary. Actually, I am a mother of, he's about 15. He'll be 15 March 19. So I take advantage to participate in those, kind of meeting anyone that I have access to. So in order to help him around, because years ago things, used to be different from now. Every time I had a chance to learn something different or whatever it is. So I know in the long way that's gonna help, help him, even me. So that's why I'm trying, to get to whatever it is.
Great. And where does Angie go to school?
He's in a Weekly Avenue, Weekly High School.
Okay. All right. Great. Well, I'm happy to meet both of you, even though it's only virtually. We've been doing a lot of that these days.
Something.
What?
No, I said I forget. He's in ninth grade.
Ninth grade. Yeah, that's great. It's great. It's a good time to start thinking about
Yes,
science, right?
definitely. Time is running quick.
Well, that's good.
Yeah.
So today I'm gonna talk to you, but I really want it to be a conversation, especially since there's so few of us. So if either one of you has a question, just stop me. I don't have a very long presentation. I was hoping it would more be an exchange, right? So, science, especially biology, has all these terms that we biologists don't pay any attention to anymore. But if there's a word that I say that you're not familiar with, just stop me and say, what's that? Because I sometimes don't remember, when I learned what, and been at it for a while. And so it can kind of get, the language can get a little, bizarre, I'd say, as we go through this. So if I say anything that you don't understand, just stop me. And if you have questions about details about anything, because of course I can't, give you. I didn't prepare all the details, but if you want to know anything in more depth, I think we have plenty of time to do that. So I'm going to share my screen here. And just put it in. So can you see this presentation mode?
Yes. Really hot and yesterday, cold, hot and
Yeah.
Oh,
there's someone else is coming in. So let's hold on just a second and meet her, Grace.
Well, that's a great presentation.
The world is an amazing place.
It is.
So, hello, Grace. How are you? Are you with us here? Can you hear us?
Mute.
Yeah. Hello. Don't you love Zoom or WebEx video conferences? So you're muted, Grace, if you want to say something. We introduced ourselves, but I'll start again and I'll tell you that my name is Kathy Royer and I'm a professor of biology at RPI. Um, and we're going to talk about extreme life on Earth and the proteins and biomolecules that are in them. So maybe you could tell us a little bit about who you are, if you can unmute. Mm. Oh, well. Let's see. Ah, there you go. Okay, well, I guess I get started, but I was telling Marie and Angie that if you have any questions or something I say, you're in the 10th grade. Hi, Grace. I just saw the chat. Nice to meet you. What high school do you go to? Do we have a 9th grader and a 10th grader? Westover High School. Okay, great. Well, welcome. So I'm going to talk a bit about the strange biology that we have here on Earth and what we can learn from it and what we can use it for. And like I said, if you have any questions, if anything I say isn't clear or you don't understand a word that I use, sometimes we use these technical words that we don't really need to use, but we don't think about. Just stop me, or I've got the chat right in front of me, so I can see it right away if you send me a message. Anybody, so that's fine. Or you can just pipe up. You can take off you can unmute and just talk to me if you want. So so we're used to really, I mean, you might think it's cold out and it's going to get colder. And so we could think of that as an extreme environment. But in fact, the Earth has many, many, many places that are much, much more extreme in terms of the physical and chemical conditions in them. So you may have seen like pictures of like Yellowstone Park or something, or even up in Saratoga, right? They have these hot springs, and then a friend of mine who's a colleague at RPI is a geomicrobiologist, and she actually goes to volcanoes and collects samples from around these really, really hot places, which are where the magma from the earth is, coming up and spouting out. And then there's some really cold places like the Arctic seas or the Antarctic seas, or even the land masses and ice flows in those regions of the planet. There's some really salty places like the Great Salt Lake, but there are other ones. The Red Sea, for example, in the Middle East, where the salt concentration is so high that the salt just comes out of solution and makes these precipitates, we call them, these blocks of salt that are just, let me get my pointer here. Where's your pointer? Right, so you can see these blocks of salt in this highly salty environment. There's really interesting places in the bottom of the ocean where there's sort of volcanoes that come up from underneath the oceanic crust, and the magma coming through those volcanoes meets the cold seawater and turns it really, really hot. And so these are called hydrothermal vents in the bottom of the ocean. And, the water coming out right here, as this really, really hot water comes out of these vents, can be up to 400 degrees Celsius, which is even more in Fahrenheit. So it can be really, really hot. And interestingly, it's also deep, right? These places are, 2, 4, 5, even 6 kilometers. So, maybe 3 miles or so underneath the ocean's surface. So they're really, really deep. And so in addition to being hot, they're also under a lot of pressure, right? All that water on top exerts pressure. And so Whatever lives there is living under pressure. There are pressures in the ocean that are 1,000 times what we experience at what we call atmospheric pressure, the pressure that we live in, as humans and other life forms on the surface of the planet. So there's some really, really strange places, but there are lots of organisms that live in these places. I didn't know, you might have heard on the news, I heard it about a week ago, they found in the Antarctic Ocean the largest group of cold water fish ever known to exist. They don't know how this happens, but anyway, there are tons and tons of fish and other organisms in these places. Because there are all these organisms that live in really extreme environments, they have to have adapted to survive in those environments. And so the molecules that make up these organisms are different from the ones that make up, say the bacteria that we try and clean off our hands when we wash our hands, or the bacteria that live in our guts. We have a lot of bacteria inside our bodies that help us digest, for example. But the molecules of life, the molecules that allow us to do all the things that we do, to get energy from food and to copy our genome and make new cells, all those things are done by molecules, and they're not the same in these organisms that live in these extreme environments. Our molecules are very fragile compared to theirs. And so that means that we could, if we knew about those molecules and how they worked, we could actually apply them in biotechnological, situations, and in particular in health science applications, but many more also. And so it's really interesting to explore these places. And here you see, for example, a ship that's outfitted to go explore. And here they've sent a robot down to a hydrothermal vent, and they're taking samples, right? It's pretty amazing. I have a colleague that I work with. He works at the Scripps Institute for Oceanography in San Diego. And he's out on a ship right now collecting samples in the Puerto Rican Trench, which is turns out right near Puerto Rico, there's a really, really deep part of the ocean. And so they've gone there to sample. So they send these robotic contraptions down to the bottom of the ocean, and then they sample, they sample the chemistry, and they sample the geology and the rocks, and then they take samples also of the life forms that they find down there. Here's a sample that's come up from inside the earth, and there's all sorts of life forms. You can see sort of pictures of the microbes that have been found in these places. This is another sort of robotic system that they dump into the ocean from a ship. You can see it's connected to the ship here to take up seawater, because there's just life floating around in the sea too. It's just everywhere. And in fact, we think that life probably originated in those hydrothermal vents a long, long time ago. So the Earth was formed about 4.5 billion years ago. Sounds like a long time. And it was hot, and it wasn't hospitable at all. And so it took a while for life to emerge from the chemicals that were floating around on Earth. And because it was also, the atmosphere wasn't the same as it is nowadays. And so the Earth was being bombarded by radiation that was very damaging. And so they think that at these hydrothermal vents, that I just showed you, the life forms would have been protected both from the radiation and the heat, and so it would have been a propitious place for chemicals to come together to start to form the very beginnings of life. And about, I don't know, that Earth was formed about 4.5 billion years ago. And less than 8 billion years later, the first organism, we think, was actually alive. We call it Luca. It's called the last universal common ancestor. So in this really simplified tree of life, you see there are animals like us, and we're mammals. There are fish and insects. There are fungi, plants. And then there are, so these are called eukaryotes because they have a nucleus in their cells. And then there are archaea, which are single-celled organisms, and all of them. And then there are bacteria, like I was telling you, the ones in our guts and stuff. And so there was about 3.7 billion years ago, an organism that we call Luca, from which all of these life forms evolved, us, birds, and various bacteria. And it's amazing that Luca actually existed only 8.8 billion years or 800 million years after the Earth formed. I find that astounding. It didn't take long for life to take hold on Earth. And then from Luca, we first got bacteria and archaea here, and then branched out into the eukaryotic organisms that came up later. So it's pretty astounding, I think, that our little Luca had just about everything in terms of molecules, all the molecules to do all the work, to get the energy, to eat, eat the food that was a little bit of food that was available, and to grow and divide and to evolve. So this talk is a lot about evolution. So if there's anything stop me about evolution if you have any questions about that, because we're really interested in how the molecules evolved to be able to function in these extreme environments. So over the past, I don't know, probably 15, 20 years, the sampling programs that are funded by, agencies like NASA and the NIH, and the Department of Energy and various, and some private institutions, these sampling programs have kind of exploded, and they've allowed scientists to take samples, like I was saying, with like these robotic things connected to ships, down at hydrothermal vents in the ocean, like here, or in taking core samples in the marine sediment, or even in the crust, the oceanic crust underneath the ocean. Right? Pressure gets really high down there. But they've also sampled in mine shafts. They take advantage of the fact that we're mining minerals or oil or natural gas or whatever all over the world, and scientists have been able to send machines down to take core samples of the rock in these places, right? And they have found lots and lots of organisms. Like these bacteria that were found. It's near a hydrothermal vent, or this really, really strange-looking fish that was found deep, deep, deep in the ocean, or this worm that was found in a mine shaft. And then life at hydrothermal vents is just teeming, actually. There's just lots and lots of life in these places. And we think that hot springs, for example, are sort of our window into this deep world, because hot springs are where, instead of this coming up, bubbling up under the ocean, it kind of bubbles up onto land, and we can have access to these strange places. And so what these people do when they get samples is they collect organisms, right, so that they can study them, but they also just collect the mud or the sediment, or they scrape they scrape a little bit off of the side of these vents, right? Um, and and then they purify out the DNA in there, and they sequence it, all the DNA. We call that metagenomic sequencing. Um, Instead of just, you may have heard a few years ago, the human genome was sequenced. It was a big deal, maybe 10 years ago. Now you can sequence a human genome with 23andMe or whatever, really quickly. But at the time, it was a huge effort to sequence the human genome. But now we can just sequence all the DNA that we can collect in an area like this hydrothermal vent, or they extract the DNA from these core samples that are basically rocks. And there's DNA in there that belongs to organisms that live in the little interstices in the rocks, unbelievably, but it's true. And so it's just created a ton of genomic and metagenomic information sequences, just tons. So if you're thinking, I tell my students, biology is drowning in data. And if you're thinking about going into biology at all in your life in the future, bioinformatics and finding new ways to pull the information that's in all these data out of them is really one of the most important things that we can train for right now, because it's actually probably easier to send these machines down to the bottom of the ocean than it is to actually get the information out of the genomic sequences and metagenomic sequences that we collect. So, what they do with this DNA is that they sequence it, and then they sequence little pieces, and they cut and paste it all together. And from that, they can figure out which organisms live in a particular environment and what kind of biochemistry and biogeochemistry is happening there. So, again, astoundingly, over 80% of the microbial biomass on the planet actually is from these very deep places, these trenches, the sediments, the Earth's crust. When you go to these places and you take samples, there aren't always a lot of organisms in each sample, but because most of the Earth is either ocean, crust, sediments, it really ends up being a lot of the microbial biomass. And microbes are we think of single-celled organisms, right? They're also eukaryotes. They're the worms and the weird fish and all that. Very strange little kind of shrimpy type beasts that live way down here in the very bottom of the ocean in the Mariana Trench. It's 11 kilometers down underneath the surface of the ocean. It's really very, very deep. And there's, 1,000 atmospheres of pressure there. So anyway, it's a very, very inhospitable place. And yet there's life down there. And what I'm interested in, so this is a picture, a schematic of a bacterial cell. And here's its DNA. There's all little molecules inside of it. This is its membrane that protects it from the exterior. And, all of these structures are made up of molecules. There are proteins and lipids. There's DNA and RNA. And we know that the molecules that exist and do these jobs in organisms from, the surface of the earth, where it's not too hot and not too cold, those molecules, if you subject them to the conditions at the bottom of the ocean or in these vents or deep in the crust, they our molecules and the molecules of the other organisms that live with us, they don't function. They get broken down, they fall apart, they unravel, they don't do their job. And so we've tried to figure out how, since extremes of pressure and temperature and acidity and salt and all that will really disrupt the biomolecules that do the work in our cells, how have the sequences of the biomolecules from these organisms in these extreme environments, how have they evolved to function? And secondly, can we harness these molecules or their just the ways that they've learned to function for applications in health science and biotechnology? And so, so this is like the fundamental molecule of life. I don't know if you've seen it before, but this is DNA. It's a schematic representation of DNA. DNA is made up of these, what we call bases that are linked together. These are all little atoms here. And there's a base called adenosine, another one called thymine, and they tend to pair up in DNA. And there's guanine and cytosine, and they pair up in DNA. And then they get all linked together in what we call the double helix. You can see it here too. And the sequences of A's and T's and G's and C's in your DNA actually end up coding the amino acid sequence of proteins. So the evolution of organisms occurs at the level of changes in the DNA sequence. Like the virus they keep talking about, the COVID-19 virus, SARS-CoV-2, all those variants of the virus that keep making our lives miserable come because actually it's RNA in the virus, but the RNA mutates. So the nucleic acid that codes for the proteins changes its sequence. So instead of a T and a C, it'll be maybe a G and a C here instead. And that allows it to evolve and escape our immune systems. Anyway, so we're interested in the evolution in these organisms. And so this is what's called the genetic code. And it shows how the sequence in the DNA is transformed into a protein sequence. So like I said, in DNA, there are 4 bases, A, G, T, and C, that, are just repeated in some sequence. You can have like A, T, G, C, T, 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 A, G, or you can change that sequence, and there are millions of base pairs in our genomes. And they code for proteins. And so the way they do that is through this thing called the genetic code. And so 3 bases that are made up of combinations of these 4 bases, so like TTT or CTT, code for one of the 20 different amino acids that make up proteins. And so there's a first base, a second base, and a third base. And you can see, for example, you just have to change the third base to code for a different amino acid. So TTT gives you phenylalanine, which is, where is it? Here it is, over here. So it has this ring off of it. But if you change that last T to an A, TTA, then it'll give you leucine, which is a different amino acid that doesn't have a ring like this one does. So it has different properties. And so the sequence of the proteins actually determines how they work, how they look and how they work. And so changes in the DNA end up giving you changes in the protein sequence. And so the way this is like a picture of a cell here, a eukaryotic cell. And there's DNA here. This is where the DNA is in the middle of the cell. And there's a machine called RNA polymerase that takes that sequence, it opens up the double-stranded DNA and it reads like you would read a book. It reads, the machine reads, it's a molecular machine, it's a protein, and it reads the sequence here. And it turns that into RNA, messenger RNA. And messenger RNA binds to another machine called the ribosome, which is made up mostly of other RNA molecules and some proteins. And that machine, along with these little guys who go and get, these purple blobs are the different amino acids here. They go and get the amino acids and they put the right amino acid to correspond to the sequence that's right in the middle of the RNA in the ribosome. And so that produces this polymer that we call a protein that has the sequence that was encoded originally in the DNA. And so, this ribosome adds more and more and more amino acids to the sequence and they get connected together so they don't come apart by the ribosome. And that polypeptide, we call it, so it's got a lot of amino acids, folds up into a structure that then does some kind of function like processes sugars so that we can get energy from sugar, for example. So many different processes in our cells that allow us to live are carried out by these proteins, and the sequences of the proteins come from the sequence of the DNA. So lately, as I said, we just have been getting massive amounts of sequences. I mean, millions and millions and millions of sequences of different, this is a sequence of the same protein, but from different organisms, right? It's just part of the sequence of that protein. And so every line is the sequence of that particular protein from a different organism. And so you can tell, for example, that here at this position, this amino acid called asparagine, which we label with an N, in this other organism turned into a histidine, H. So it's a different amino acid because there was a mutation in the DNA. And all these organisms have H here until you get to the organisms that end up having an S there, which is a serine, a different amino acid. And so, you can see that there are changes. There are many amino acids that are the same or similar, but many that change. And so we've got tons of this information. So if you're thinking about what you're going to do when you go into your professional life, trying to understand why these changes occur is really a huge challenge to us now. Because if we could understand why, then we could make proteins to do basically whatever we want. So anyway, so we're getting bombarded with all this data. And we don't know how these sequences really evolved, which of the changes in the amino acid were important to allow them to function in these strange places at high or low temperature, high pressure, low pH, acidic environment, high salt concentration, right? We don't know how, we know that the environment on Earth shapes the genomes of the organisms, the sequences, and the proteins, but we don't know how. That's a a question that's open. And it's a really important question, basically, for understanding life on Earth, on how it has evolved and may continue to evolve with changing environments, which are changing with climate change quite a lot. It also helps us to understand what we call geobiochemical cycles, right? So tons, billions of tons of carbon and sulfur and nitrogen are cycled on the planet from one place to another in different forms, going up into the atmosphere, coming back down to Earth, going into the water. It's just, and a lot of those cycles are driven by these microbes that I've been talking about. You may have heard about cows causing climate change because when they poop, they give off methane. And methane goes up into the air. And that methane is actually produced by bacteria that are in the guts of the cows, right? So the cows eat the grass, and the bacteria help the cows to digest, just like the bacteria in our guts do. And then, we get this methane gas from that. And so we're trying to capture the methane from cow dung. Anyway, so there are really, really real implications for our changing climate. In understanding how these proteins work. I mean, if we could get a protein that could, transform methane into some other non-toxic compound, then that would be great. It would, we could make a factories that biological factories that just transform methane to get it out of the atmosphere. And, if we understand how these sequences evolved to make the proteins function under these different conditions, we might be able to use that information to make designer proteins to do whatever we want in biotechnological applications, right? Instead of making a lot of chemicals that we use in drugs or in other contexts by organic chemistry with lots of nasty solvents and stuff. We might be able to use bacteria. People are using bacteria to make some of these compounds. And we could probably even use proteins themselves to make these compounds in bioprotein reactors. So there are a lot of biotechnological applications. And we could actually discover in these genomes new enzymes those are proteins that carry out chemistry that's useful to us. There may be new enzymes that have properties that we could put to use in all sorts of applications. So biotechnologically speaking, mining the information that's in these really extreme environments could be. So I'm going to tell you about one enzyme, which is one of the oldest enzymes in the world. It's an enzyme that takes sulfate that you see sometimes in these acidic hot springs. There's a lot of it down in those hydrothermal vents, right? It comes up from the Earth's core. And this enzyme takes its form when it's been reduced a little bit to sulfite and turns it into hydrogen sulfide. And you may know about hydrogen sulfide because it's what makes the hot springs up at Saratoga smell like rotten eggs. When you smell rotten eggs, it's because of hydrogen sulfide that's being made. So that's why hot springs oftentimes smell like rotten eggs. And this is just a picture of the protein, a cartoon of the protein. It's really pretty, I think. It has lots of different parts. It's got, places, extra molecules in it, like this red one or these yellow ones that help electrons move around inside the protein. And this enzyme, that's probably one of the oldest. It's thought to be 3.5 billion years old. So right when life was coming up on Earth, this enzyme actually metabolizes billions of tons of sulfur compounds every year on Earth. And it's really important for the biogeochemical cycle of sulfur on the planet. And it has also links because of that to carbon cycling on the planet. So implications for climate change and environmental change on the planet. So it's really important. Yeah,
Oh, can I ask a question?
absolutely.
Uh, dissimilatory sulfide test. What
Mhm.
is the effect of that on human, on Earth, like me and you, whatever?
Well, it's found in bacteria, which are called sulfate-reducing organisms, and they're everywhere, all over the place, and so it cycles those organisms all over the planet are responsible for moving sulfite, from or sulfur from this form sulfate to this form sulfide, right? And that is linked that in the bacteria, it's also linked to carbon transformation. And so the way the tons and tons and tons of sulfur that gets processed by this enzyme in all these organisms is linked to us is through the carbon cycle, because there's a link between the sulfur cycle and the carbon cycle. And so these enzymes, they actually, when they do this, they allow this reaction here allows the organisms to extract energy and use that energy for metabolizing carbon also in the organism. So the way it affects us is through the carbon cycle.
All right.
Mostly.
Thank you.
You're welcome. Yeah. And so global warming is how it,
It is. Yeah.
so either, I mean, it's impacting how global warming is happening. It's not necessarily promoting global warming. It's probably
It's
actually,
all
yeah,
right.
helping to stop it.
Okay,
Anyway,
thank you very much. It's clear
you're
now
welcome.
for me.
But it's really, really billions of tons. It's an amazing amount. These are tiny little organisms. They're about uh, 1/1000th of a millimeter long, right? These organisms.
Wow.
They're really little. Uh,
Extremely long.
really small. But they have all these molecules. Molecules are even smaller. So, they have tons of them in them, and there are tons of these organisms, right?
Yeah.
So they can make a big difference.
Okay.
So one of the things that bioinformatics people do is they take all the sequences uh, from all these little things you can't read here are the names of different organisms, right? And they take the sequence of the enzyme from these different organisms, like I showed you that list there, and they do a statistical analysis to figure out uh, what was the oldest one of those enzymes and how did it evolve? So this is called a phylogenetic tree. And so the original organism was here. We don't have the sequence of that one, of course, because it was too long ago. But the statistic says that that one branched into two branches here, and then this branch branched into two branches, and then branched into two branches here. And so one of the oldest of these organisms is this one here, this first one, because it's only 1, 2 steps away from the very beginning, right? According to the sequences. And so, you know, how is this really old one different from one of these ones at the end, the newer ones, right? Um, and here, this phylogenetic tree has been colored for this first outer circle is the environmental, you know, uh, um, places, conditions where the organism lives, right? So the purple ones, like there's one here, live where there's a lot of salt, hypersaline, right? These blue ones here, this one, for example, is freshwater, and this one's freshwater. Sorry, these darker blue ones are marine. Red is thermophilic, in like the hydrothermal vents, right? And black is in the subsurface. Oh, sorry, I'm down here. So black is the subsurface here. And then the second circle is the taxonomic group. So like all these red ones here at the very end, these are from a group of bacteria called Delta proteobacteria. You can see that there's some who are archaeota. So they're from the archaea, the other realm of life, right? There was the bacteria, and then the archaea, and then the eukaryotes, right? And there's euryarchaeota too, which are, let's see, these guys here, euryarchaeota. So anyway, the blue ones, yeah, these are euryarchaeota. So there are all sorts of different proteins. And it's interesting, we found out that after this branch here, all these are proteins that run the reaction backwards. They take sulfide and turn it into sulfide to go. So they actually do just the opposite. So they take this compound and turn it into this one instead of going this way. We don't know how they evolved to do that, but all these organisms are ones that turn the system around. So you can learn a lot about how things evolved and how an environment might have impacted the change in sequences, and how taxonomy, so which group it belongs to, might have done that. So phylogenetic analysis of these sequences is really important. That's one kind of bioinformatics. But there's another bioinformatics, the kind that I do, where we try and figure out what were the important changes, because evolution is random, like the COVID SARS-CoV-2 is just randomly mutating all the time. And only some of those mutations are really useful for it, right? Like the Delta mutation. And others are just kind of, they go along for the ride. They mutated, but it didn't really matter. It didn't hurt the organism, didn't help the organism. So they're there, but they don't do much. And when you just read sequences like this, it's really hard to tell whether this D was important or should it be an E, or whether this I was important or should it be a K, right? We don't, we can't just read. We have a book, basically. We have a ton of books. We have libraries, basically, in these sequences, but we don't know how to read them, really. We don't know. We know what the amino acids are, but we don't know how to extract what's important information. And so that's really a challenge to the future in biology, is to continually increase the amount of important information we can pull from these sequences. And so one of the things so sometimes you see that an amino acid, this is a cysteine, a C, is always conserved in all the sequences. If you align, 10,000 sequences, it's always a cysteine there. So that one's really important for the function, because that one, when it mutated, the organism didn't survive. And so we don't have a record of it anymore. The only records we have are ones with that being cysteine. So it stayed there. It was important. So that we know, if a position is conserved, it's doing something. We don't necessarily know what, but it's doing something. And then there are positions, if you do a more sophisticated statistical analysis of these sequences, you can figure out that there can be positions that if the first one, this R changes, say, to a K, then the D has to change to an E. And So we call those co-evolved positions. So only when both of them are mutated and in a special way does the mutation, or do the mutations get kept, right? The organism can survive. It's viable. And so for example, we know this amino acid arginine is positively charged, and we know this one aspartate has a negative charge, and positives and negatives attract each other, right? And so that probably means that there's an interaction between those two positions. But if you change this R, to a K, it's still positively charged, and this is an E, so it's negatively charged. But this is a smaller positive charge, and this is a larger negative charge, right? So they have to move together, right? You could also sometimes probably see if this one is changed to a D, then this one has to be changed to an R to keep that positive negative interaction. You just turn it around the other way. And so these co-evolved positions often dictate interactions inside the protein structure, like the structure that I showed you here, right? So there's, say, an interaction right here between this purple part and this blue part, or between this purple part and this pink part. And so those interactions are important. If they're important for the protein, if you mutate one of the residues in the interaction, you have to mutate the other one too in order to keep the interaction. And so people have used this statistical coevolution to actually predict the structure of proteins for whom we don't have an experimental structure. So, and sometimes these interactions predict, these co-evolved residues predict interactions within the same chain of the protein, but sometimes two chains can interact, two different genes can interact, and these co-evolved residues between genes sometimes show us that where that interaction occurs in the complex. So you can get a lot of information about the structure of the protein from the statistical analysis of the sequences. And so we did this for our DSR protein, and we found actually a whole little series of co-evolving residues in the protein. You can see them here, these blue ones. And what they do is they show a pathway between these two red molecules here, and that's where the chemistry occurs in these red molecules. Right here is where DSR changes sulfite to sulfide, and there are two places where it can do it in its structure, and apparently they have to talk to each other, those two places in the structure. And so you can get information about not just how the protein is built, from these co-evolving sequences, but also about how the protein works, actually. And that's what we want to find that out, because we want to be able to build new proteins that work better, right? Make them up. So. And this is just a picture of a calculation I did. Another thing that people can do in here in bioinformatics is you can do calculations on the dynamics of your system, because proteins, when they do all those chemical reactions, they have to move, and they have to move in certain ways. And so I was able to calculate, right, how this protein may be moving, right, and maybe helping the electrons get around and do the chemistry. So another amazing thing that's happened over the last couple of years is this Google is using artificial intelligence to be able to predict the structures of proteins. And so I used their algorithm to predict the structure of that oldest DSR we didn't have. So that's another avenue for research is predicting structures. And so, like I said, the proteins from these environments have important biotechnological applications. And I'm just going to show you one. It's from a protein from a bacterium called Thermus aquaticus, which was found in a hot spring in Yellowstone Park quite a while ago. So we call it TAQ or TAC polymerase. And it does what's called a polymerase chain reaction. And that's the basis for the COVID-19 polymerase chain reaction, PCR test that you probably heard about. And so you put it in the tube along with some coronavirus RNA that you get out of your nose. What happens is the TAC polymerase, because it's from a thermophile, it lives under hot conditions. There are these protecting molecules that make it inactive. But if you heat it up, you kill the protecting molecules, but TAC doesn't care because it's happy when it's hot because it comes from a thermophile. And so now it's active and it takes your coronavirus RNA that you've transcribed into DNA, you went backwards. It takes that DNA and it just makes more and more and more of it. You just go through these cycles where you heat everything up. The strands come apart. The TAC polymerase polymerizes it like this, and then you heat it up again, and TAC does it again after you cool it. And so you can do this hundreds of times. And because TAC polymerase comes from a thermophile, it doesn't care if you're heating it up. It's just as happy to keep making more and more and more and more copies. And so it makes so many copies that they're really, really easy to read then. And that's why these PCR tests are so sensitive. They'll always tell you that they're more sensitive than the antibody tests, and it's because of this cycle of heating and cooling that the active enzyme can withstand because it comes from a thermophile. So there are lots of possibilities in those extreme environments to find enzymes like this in order to do biotechnological things like the PCR chain reaction to detect SARS-CoV-2. So I'll just finish by saying that I've been working with high school students online. I call them my extreme team. We use bioinformatics and to learn about protein chemistry and to model structures and to explore evolution. And so as you go through high school, if you're interested in doing this, just remember who I am, and you can contact me and you can be part of an extreme team that studies these structures. So I'm done. If you have any questions you might still lingering about proteins and how they evolve and what they do, I'm happy to.
Thank you very much. That was
Well, thanks for coming. I hope Angie will become a biologist. Life is really interesting.
He's thinking about it. He is thinking about it.
Yeah, well, you've got plenty of time to think, of course, but I hope that crosses your mind.
It's a little bit down.
I'm sorry, I can't hear you very well. I think your microphone is.
Wow. Let me see. Well, now can you hear me clear?
Yeah, now it's good. That's good.
Okay, maybe my volume was too low. No, I said he's thinking about it.
He's thinking about it. Well, if you have any questions about it, I mean, not just about what I said here, but about biology or biology at RPI or anything like that, don't hesitate to contact me.
It's a nice subject.
It is. It's just super interesting, I think.
It is. Yeah, I like biology, but
well, I hope you learned something. It was good to meet you.
Good to meet you too. Have a nice afternoon. Thank you very
Yeah,
much.
you too. Both of you. Thanks a
Bye bye.
lot.
You too. Enjoy the rest of your day.
Okay.
Bye.
Bye.